be talking about cellular respiration and the first phase, which is called glycolysis. Glycolysis occurs in the cytoplasm outside of the mitochondria and is the breakdown of glucose to two molecules of pyruvate. Energy is invested to activate glucose. 2 ATP are gained, and oxidation results in NADH, which will be used later for additional ATP production. So this process is divided into two steps: the energy investment step and the energy harvesting step. For the energy investment step, when some ATP is used to begin the reactions, 2 ATP transfer phosphate groups into substrates, and 2 ADP plus P result. Those break down into 2 G3P. For the second step, the energy harvesting, electrons and hydrogen ions are removed from the substrates and captured by NAD, producing 2 NADH. Then there is a reduction in H2O. As the reaction proceeds, the energized phosphate groups on the intermediates are used to synthesize 4 ATP. This is accomplished by a process called substrate-level ATP synthesis. How the reaction proceeds from here is determined by whether oxygen is present. If oxygen is available, pyruvate, the end product of glycolysis, enters the aerobic reaction within the mitochondria, where it is used to generate more ATP. And this leads into the next phase, which is preparatory reaction, and we'll talk about that in the next video. Next, we're going to be talking about preparatory reaction and the cellular respiration. So for the first step, pyruvate is oxidized and a CO2 molecule is given off. This is part of the CO2 that we breathe out. Next, NAD accepts the electrons in a hydrogen ions, forming an NADH. The product, a 2-carbon acetyl group, is attached to a coenzyme A, forming acetyl-CoA. Therefore, per glucose molecule, the outputs are 2 CO2, 2 NADH, and 2 acetyl-CoA. And then we move on to the citric acid cycle. The citric acid cycle, it is a cyclical metabolic pathway located in the matrix of the mitochondria. For the first step, which is the second step in the full process, 2-carbon acetyl group combines with a 4-carbon molecule to produce a citric acid, which is a 6-carbon molecule. For the next step, twice oxidation reactions produce NaH and CO2 is then released. Next, the loss of the 2 CO2 results in a new 4-carbon molecule. Then ADP and P combine to create the ATP. ATP is produced by the substrate level ATP synthesis. final step of the citric acid cycle, additional oxidation reactions produce another NADH and an FADH2 and regenerate the original 4-carbon molecule. And this is what the whole cycle all put together would look like. You have the first step in black, second step in pink, third step in green, and fourth step in blue. This is the process of the electron transport chain. The electron transport chain is located in the cristae of the mitochondria. It is a series of carriers that passes electrons from one to another. NADH and FADH2 deliver electrons to the chain. The hydrogen atoms attach to NADH and FADH2, and they consist of an electron and a hydrogen ion. The carriers of the electron transport chain accept only E negative, which is the electron, and not the hydrogen atom. The high energy electrons are being transferred by NADH and FADH2 from glycolysis, the preparatory reactions, and the citric acid cycle. The electron transport chain is a series of redox reactions that remove the high energy electrons from NADH and FADH2. As these reactions occur, energy is released and captured for ATP production. The final acceptor of electrons is oxygen, or O2. Once the oxygen is accepts the electrons, it combines with hydrogen to form a water molecule, so H2O, which is one of the end products of cellular respiration. When each NADH delivers electrons to the first carrier of the electron transport chain, enough energy has been captured by the time the electrons are received by O2 to permit the production of 3 ATP molecules. When each FADH2 delivers electrons to the electron transport chain, only 2 ATP are produced. Once NADH has delivered electrons to the electron transport chain, NAD is regenerated and can be used again. In the same manner, FAD is regenerated and also can be used again. Just as in photosynthesis, the hydrogen gradient contains a large amount of stored energy that can be used to drive forward ATP synthesis. The cristae of the mitochondria contains an ATP synthase complex that allows hydrogen to return to the matrix. The flow of hydrogen through the ATP synthase complex brings about a conformational change, which causes the enzyme ATP synthase to synthesize ATP from ADP plus P. The ATP leaves the matrix by way of the channel protein. This ATP remains in the cell and is used for cellular work.